Hi, everyone. Well, I'm going to share my screen very quickly. Okay. Okay, can you see my screen?
Looks good.
Okay, thank you. Well, hi, everyone. As you mentioned, I'm going to introduce a little bit all the workflows that are available in CartograPlant, and I'm going to give you a little bit of background about why these workflows are, why we have chosen these specific workflows for CartograPlant. So, well, as Jill mentioned in the introduction, all these analyses are possible thanks to a Galaxy instance. And our main objectives are basically to perform association mapping analysis, that is, associate the genotype with the phenotypes, landscape genomics with associate the genotype with environmental information, population genomics, and also the meta-analysis of these two analyses, association mapping and landscape genomics. The main objective is just to take advantage of one of the main strengths of CartograPlant, which is the integration of different data types coming from different studies, and the information of this wide diversity of data types, of course, the environmental data, phenotypic data, and genotypic data. So I'm going to show a little bit what is basically a genome-wide association analysis. I'm going to give a brief description of that. So this is basically an approach used in genetics research that associates genetic variations with traits of interest, with phenotypic traits of interest. So in this case, the genetic markers, the genetic variations act as predictor variable, and the phenotypes as a response variable. So this association is made by the use of a model, normally a regression model. So for instance, this association allows to obtain genes that are related with traits of interest. Let's say, for instance, that we are looking for candidate genes of resistance to drought. We can just find them by using this kind of associations. And landscape genomics is very similar to genome-wide association analysis, but in this case, we are associating the environmental information with the genotypes of interest. So again, following the same example, if we are looking for genetic variants that confer drought resilience, we can just associate the genotypic markers with drought environmental conditions, with dry environmental conditions, in order to find potentially locally adapted genes to these conditions. So now the question is, why using these two approaches together? Because it seems a little bit redundant, right? Because they can answer very similar questions. And the answer is that the use of these two approaches together, what it does is help us uncover patterns that cannot be detected using one approach alone, because both approaches support each other and enhances the association power this way. This is why we include both approaches in CartograPlant. Now, this analysis can have several factors that can affect the accuracy of the results. They can be classified into four, basically: the genotyping errors, the existence of missing SNPs, the existence of population structure, or kinship in our populations. So regarding genotyping errors are basically, it occurs when the observed genotype does not correspond with the true genotype. And it can happen at any point in the experimental workflow. So during the sample preparation, for instance, or the library preparation, or the sequencing. And in order to deal with these potential genotyping errors, CartograPlant provides a lot of tools for SNP quality filtering. So the objective of this SNP quality filtering is basically to remove markers and individuals that likely contain genotypic errors. For instance, those containing a high percentage of missing data or low quality SNPs. Another potential problem is the existence of missing SNPs, or even after performing this data quality filtering, some missing SNPs may remain in your data set. And these missing SNPs reduce the statistical power of our association analysis. So in order to deal with this potential issue, CartograPlant offers a workflow to impute these lacking SNPs. This imputation or inference of the lacking SNPs can be done by two different approaches. One of the approaches is by using a reference panel of SNPs. It's a panel that containing high density SNPs and obtaining the same species, in the same model species. But this approach is not suitable for non-model species because it requires a high quality reference genome, and it is not always available for non-model species. For model species, yes, but not for non-model species. And one of the objectives of CartograPlant is to provide tools for non-model species as well, not only for model species, but for non-models, which is one of the main gaps that we have nowadays in the majority of plant molecular databases. So in order to fill this gap, we are providing a SNP imputation workflow that makes use of another approach that is suitable for non-model species. And it is suitable for non-model species because it uses linkage disequilibrium. And it doesn't require any high quality reference genome or a high quality or high dense reference panel of SNPs. So I'm going to explain very briefly what linkage disequilibrium is. So this is basically a non-random association between two or more loci in the population, or in other words, that two or more loci tend to be inherited together. And it depends on the distance between the loci. So the closer the loci are, the stronger is the linkage disequilibrium. And it is very useful for our analysis because it's the basis of the identification of causative or locally adapted SNPs, because sometimes the genotype SNP is not the truly causative SNP, is not truly linked to the phenotype of interest or the environmental data of interest, but it is in linkage disequilibrium with the truly causative SNP. It is also useful for this SNP imputation because sometimes some of the SNPs are lacking, but they are in linkage disequilibrium with some of the SNPs available in our data set, and we can infer them based on that. So in order to do this imputation of SNPs, we use in CartograPlant a program that is called LinkImputer. So this program basically does two types of analysis. On the one hand, it performs SNP quality filtering. In particular, it provides different combinations of different quality filtering thresholds, because sometimes the quality filtering thresholds are very dependent on the study. So there is not a standard value, for instance, for a quality score of a SNP or for a percentage of missing data in a loci. It depends on the data set. So it is tricky for users to decide which one is best for your data. And this program allows to perform different combinations of candidate thresholds provided by the user, and provides this output showing the number of SNPs retained, the number of individuals, and the accuracy and the correlation of the results. And this way the users can select the combination of thresholds more suitable for your data set. So after doing this kind of data quality filtering, this program also performs this imputation of the lacking SNPs using the linkage disequilibrium approach that again is suitable for non-model organisms. Another factor that can affect the accuracy of the results of this association analysis, landscape genomics and genome-wide association analysis, is the existence of population structure or kinship in your populations. So any kind of structure can lead to the so-called spurious associations or false positives, because they can act as a confounding variable, which is a variable that correlates with both the predictor variable and the response variable. So it is necessary to account for this kind of structures in your analysis, in your association analysis, in order to avoid these false positives. One way to deal with that is by using linear mixed models. So in order to deal with that, we provide in CartograPlant a workflow that performs genome-wide association analysis using a program that it is called Emacs, that makes use of a linear mixed model. And these are very useful models for this kind of analysis, because it allows to correct by a wide range of sample structures, including population stratification and kinship. So it makes use of an internally calculated kinship matrix. So this program calculates the kinship matrix, and an externally calculated population structure. So this population structure has to be calculated externally to this program. And to do so, CartograPlant also provides a population structure workflow. In particular, it makes use of the program FAST Structure, that is a Bayesian approach to perform population structure analysis. And since Emacs is a frequentist model, it works with p-value. It provides p-values for its SNPs. It is also necessary to perform a multiple testing correction. And so we also provide a false discovery rate workflow in CartograPlant to this end. And finally, for studies containing common garden data, it is also necessary to calculate the best linear unbiased predictors for the traits, in case that we are using data from common garden, of course, in order to standardize the data that come from different locations, for instance, or years. And this is the reason why we are also working in provide a workflow for this calculation. And finally, for landscape genomics analysis, we provide a program that makes use of a Bayesian approach, in this case, that also correct by these structures these potential structures in our data set. So it basically estimates the empirical pattern of covariance, and it uses as a node model to test the individual SNPs. And since this approach is Bayesian, we don't need a multiple testing correction for this approach. And finally, CartograPlant also provides tools for meta-analysis. So the meta-analysis is basically a set of methods that allows the quantitative combination of data from multiple studies. So let's say that we have 2 studies, and we can calculate in both of them either the genome-wide association analysis or landscape genomic analysis. And as a result, we obtain a document with the summary statistics of this analysis. So the meta-analysis, what it does is get the summary statistics from these 2 studies and analyze them together. And this is very powerful because this meta-analysis can improve the power to detect the association signals, because it increases the sample size and also the number of SNPs analyzed, because it combines data from different studies. This meta-analysis is possible in CartograPlant because of the metadata collection using ontologies and standards that allows the integration of different data sets from different studies. And to do so, it makes use of the Plink program. There are a lot of programs to perform meta-analysis, but CartograPlant makes use of Plink. And another thing to consider in order to perform meta-analysis is that the SNPs have to be mapped against the same version of the reference genome. So as you know, the reference genome are updated almost constantly in biological research. And sometimes the SNPs from the different studies are mapped against different versions of this reference genome. So they are not comparable. So in order to make them comparable to perform a meta-analysis, we have first to map them against the same version of the reference genome. This is why we provide a SNP remapping workflow in CartograPlant in order to have them map against the same version and be able to perform meta-analysis. And this is basically an overview of all the workflows available in CartograPlant that now, I hope that it makes more sense now after explaining all the background, all the reasons why we have these workflows in CartograPlant available. So basically, CartograPlant integrates phenotypic, genotypic, and environmental data, thanks to this metadata collection using ontologies and standards. So regarding the genotypic data, we've performed a SNP quality filtering first in order to avoid genotyping errors, a SNP imputation in order to infer the lacking SNPs. This SNP remapping is necessary for meta-analysis, as I explained. Also, we provide a population structure and kinship workflow in order to correct by population structure in the association mapping and landscape genomics analysis. And finally, by using the summary statistics of these 2 workflows, we can perform meta-analysis. So as a conclusion, CartograPlant provides flexible analytical workflows that can facilitate a diversity of biological questions and also take advantage of one of the main strengths of CartograPlant, which is the integration of a diversity of data types from different studies and also this metadata collection using ontologies and standards. And finally, I would like to thank all the members of this project that without their hard work, it wouldn't be possible. And of course, to you all for your attention.
Thank you, Irene. That's perfect. So that's kind of our very, very intensive crash course on sort of population genomics workflows within CartograPlant. And now I'm going to try to kind of pull this together by showing you a little bit of what we have on the CartograPlant side to start to search and work with these data sets to perform that analysis. So to do that, I'm actually going to go over to our Git where we have a lot of this information sort of set up as screenshots so you can follow it as a tutorial. Again, the slides that you just saw and the Git links are all in the agenda if you'd like to follow along or look at these things afterwards. And so in the beginning, I went through in kind of great detail how the interface works, how you search trees, how you load layers, all that good stuff. And what I want to show is if you're about to perform an analysis, we're going to kind of jump back to that part here. You're logged into your account with CartograPlant, which you've created using our handy guide link. Once you have that account, you can now perform any type of search, as I mentioned, by species, by phenotype, by study. And in the example that I'm showing here in this screenshot under filters, this is where you can start to conduct your searches. We've decided to search by study title, and we're actually searching for two different studies that we can use for a meta-analysis in CartograPlant. And so we set our first study title equal to whatever we're looking for, and those study titles are actually available in the dropdown, so we don't need to know exactly what they are. And then we put in another study title here, and we get that information to both appear on the map. And then there's also now this handy button that comes up that allows us to select all of the plants that are represented by those two studies. And because we have logged into our account, we can now go up and we have access to the analysis option. And all of the plants represented by those two studies are now available for our analysis. And so once we go into the analysis tab, we click up there. We now get to set up what type of analysis we'd like to conduct. So we're going to call this analysis serotinib because this is a study where the primary phenotypes that were measured, we're looking at serotinase traits. And we're actually going to select the analysis type as being genotype by phenotype by environment. And essentially what this does is it sets us up with the workflows that are appropriate and the filters that are appropriate for this analysis. And so you have the option to do genotype by phenotype, you could do genotype by environment. And so as long as the data supports it, the studies that you brought in actually have this type of data, then this will be available to you. So as you move through the analysis panel, what you'll see here at the top is you have a series of tabs, and as you work through working with the data sets you brought in, you have the ability to kind of visualize and filter this data in different ways to prepare it for your analysis. So essentially, we're going to select an analysis account, and you're also going to select a workspace. And so you can create as many workspaces as you need and save them within your account for the different types of analyses you're trying to conduct. So say we're going to load up our serotinib, sorry, one moment. We're going to load up our serotinib option, and now we can see that in the analysis panel. So when we go to the next step, one of the first things we want to do is actually look at our trait data. And so in this particular set of studies that we've selected, and again, we have two studies that are selected in this interface. We have the phenotypes listed as the number of closed cones. So this is a study on conifers. We have the number of open cones, and we have 561 measures for both of those phenotypic traits. And then we also have the percentage of serotinase cones, and we have 760 measures on that final trait. And what we say is that we want to look at the overlap with all studies. And this is a really cool feature in CartograPlant because essentially, because we've taken in all of the data from these studies and tagged it with ontologies initially, that the system is able to tell us which phenotypes are actually represented by these two independent studies and allow us to select, okay, I want to get all the traits that actually overlap with those. And from there, we're able to see that that is just the percent of serotinase cones. And what the system actually will do is then plot that phenotype for you automatically. And what you can see is we have a lot of values on one end and a pretty long tail. And if you wanted to, say, adjust the phenotypes that you were putting into your analysis, you now have an interactive visualization that will allow you to modify those thresholds based on visualizing your traits and actually decide that you want to, say you can kind of swap these over, you could remove the tails, you could keep everything, but you have an opportunity to sort of look at that data and decide what you want to move forward with. And again, this is looking at the overlap between those two studies that we selected initially. And you could have any number of overlapping, studies here. So if, say, you have 5 or 3 studies that you're looking at, we could look at the overlap between all of those. And the system will also tell you when there's no overlap across those, and you might want to only use a subset of the studies that you've selected. So moving then to the next screen, I wanted to give you also an example of where you might have more than one phenotype that's overlapping. So in this example above, we just had one trait, and that was the percentage of serotinous cones. But we've also included an example here if you've got more than one phenotype that's overlapping across the studies. In this case, diameter of breast height, and then plant height. These are 2 larger studies, so we have tens of thousands of phenotypes associated with these 2 studies. You could do the same thing where you adjust the thresholds based on each phenotype independently. But one feature that I wanted to show here is that you can also look at the relationship between those traits. And that can also be another factor to determine whether you proceed with both traits or whether you actually just use one in your analysis. And so I wanted to show this view as well, just because that's an example of looking at the interaction between those traits as well. So going back to our first study, which just has the percentage of serotinous cones as our overlapping trait, we've selected that. We've done a little adjustment on our thresholds for the traits. Then we move into being able to filter genotypic data. And so at this stage, when we talk about filtering genotypic data, we're simply asking the question of how many markers are overlapping across the studies that are selected. Again, we've got 2 studies that we're talking about here, and you can see from the Venn diagram that there's a total of 257 markers that are overlapping between the 2 studies. And this little summary here gives you a nice quick view of what we have independent to each study, as well as what's overlapping. And you can see that you can put a checkbox next to the data set that you want to proceed with to the next step. And so that's what you see available here. And so as we move to the next step, if you have selected that you want to do a genotype, phenotype, and environment study, you will also have the option to filter by environmental metrics. So now this is one of the really nice features of CartograPlant, because all of our georeferenced plants that are in the database have environmental data associated with them for almost every layer that we have loaded. As long as that layer covers the region that those plants are located in, then we can pull the metrics out at those locations and use it in our analysis. And once you get into this view, and basically at this point, you've now said, okay, I'm going to be working with 257 markers. I'm going to be working with one phenotype for my analysis, which was the one that overlapped for percent serotinous cones. And I've adjusted my thresholds for my phenotype. So I have just the phenotype values I want, just the individuals associated with those 257 genotypes. And now I'm going to pull in the environmental metrics for the individuals associated with that data in the study. And here, essentially, you have the option to expand out all the layers that we hold and select the specific metrics that you want to pull in and associate with the selected plants. And so here's just an example of being able to kind of expand this long list of layers that we have, select specific environmental metrics that you want. So say temperature related data, and then you will generate that data. And this can take a little bit as we're pulling it from our very big database, but it's letting you know how that's going, and you can pull that information down. So at this stage, you now have overlapping genotypes across 2 studies, a single overlapping phenotype across those 2 studies, the ability to adjust that, and you have all the individuals that encompass that data. And so now you can move into sort of traditional filtering for GWAS as we move through. Irene kind of touched on this a bit in terms of what we need to do. before you can run into your analysis. So these are things like filtering by SNPs, filtering by individual. So we're basically looking at how to accommodate poor quality SNP data or missing data across our studies. And so when you move into this workflow here, you will have the ability to select what kind of filters you want to apply, and then also visualize what that data looks like. And so essentially you're doing all of this work within the Filter by SNPs tab. And I recommend that this is something that you kind of dive into after you've gathered all of your phenotype and your environmental data that you plan to work with. Now go back and start looking at and filtering based on the SNP data. And so the SNP options that we have are things like minor allele count, minimum quality, minimum reads per marker, percent missingness per marker, and minor allele frequency. And so those are some of the filters that you can apply. And you have the option to apply all or some of these filters. We also provide sort of recommended thresholds for what would make sense for the data, but the user really has the ability to modify those as well. And so as we move through, we have the same setup where we're working with an interactive histogram so that you could just sort of slide those bars around to adjust the data, and you wouldn't, and that's going to be often an easier way to get a view of what it looks like for data missingness, right? By individual or by marker type, for example. And so that's where you'll have the ability to adjust that information. And so that's kind of where everything leaves off in terms of the side of being able to do your SNP filtering. So you've got everything filtered by traits, by markers, by kind of values for your phenotypes, adding in your environmental data where available, and then filtering your SNP data. And then you're ready to move into the workflows as Irene talked about, which is relating to starting with imputation, for example, and moving through to GWAS. And so now I'm probably going to have Irene just do a kind of a brief, roughly 10-minute overview of what those next steps look like in her Git, so that you guys have all of that, since we're a little bit we're a little bit over time. But I think it's important just to see what those workflows are and how you would interact with those. So I'm going to go ahead and stop here and then let her show you the next steps. Now she's going to be showing this with two other studies that have more data associated with them. And she's going to share this in Git as well, so you can see those steps.
Okay, thanks, Jill. So I'm going to share my screen again. Okay, this is the tutorial that can be can be found in the agenda. So in this tutorial, all the workflows that I explain in the slides are available and explaining in further detail. But I'm going to show you the how the interface looks like. So after filtering the traits, the genotypic data by SNPs, by individual, etc., then you can just reach the last tab that says Run Analysis in order to perform data analysis. So the first step is to select the workflow you want to use for your data analysis. I'm going to show the genome-wide association analysis because it's the only workflow that is fully available right now, but the rest of the workflows will be available very, very soon. So first of all, as I mentioned in the presentation, you first have to impute the lacking SNPs in order to enhance the accuracy of the statistical association power of the analysis. And to this end, we have available the program link imputer that performs, on the one hand, it performs the SNP quality filtering as well. So it combines different, it performs different combinations of thresholds in order to decide which one is more suitable for your data set in case of you still have doubts after all these nice visuals provided by CartograPlant. So to do so, it is necessary to provide a configuration file, as you can see here, and a VCF file containing the SNPs, of course. So these SNPs have been filtered in previous steps, and the configuration file has to be provided externally. So you can design it in your laptop, and the information about this configuration file is provided here in the tutorial. So you can go here to data quality filtering, and let me see. And if you scroll down, you can see, you can find how the configuration file should look like. There you basically provide the different thresholds that you want to test here. The rest can be leave as default. So you can just provide here the different thresholds you want to test and upload it here. So by clicking here in this tab here, you can just find your file. I'm not going to upload it because it takes time, but you can just select it. And also the SNPs, you can find it here, the SNPs that you have selected in previous steps, and click Initiate Analysis Job. It will provide you as a, It will provide you like two different outputs, one XML file that you can use it in the further step, and a .dat file that I'm going to show you very quickly. So this file, this output file provides you the different cases that has been tested in LinkImputer, and you can select the case that is more suitable for your data. For instance, in this case, the case 5 will be the most suitable one because it provides you the higher number of individuals, the higher number of SNPs with a high accuracy and correlation. So we will select it in the following step in LinkImputer. So here you can select the step 2 of LinkImputer, and again provide this XML file, which is the output of the previous step, the VCF file containing the SNPs, and also type the case you want to use in order to perform the SNP quality filtering. And also this step will also perform the imputation of the lacking SNPs. So the SNP quality filtering if you want to, and the imputation of the lacking SNPs. Once you have performed this, you can go to the genome-wide association analysis. So you will obtain as an output a VCF file containing your filter SNPs and also the imputed SNPs. And you can go to the genome-wide association analysis, which is this workflow here, with Emacs. That, as I have mentioned during the presentation, is a linear mixed model that allows to correct by different population structures. So here again, you can provide your SNPs, which is the output of the previous step, the filter SNPs and the imputed SNPs. Also, you have to provide a VCF file with the SNPs filtered by linkage disequilibrium, which this workflow is also available in CartograPlant. So this is because Emacs, as I mentioned in the presentation, performs the kinship analysis internally, but it assumes that the SNPs are not linked by linkage disequilibrium. So you have to filter them previously by linkage disequilibrium. This workflow is also available in CartograPlant. Also, the phenotypic data filtered in the previous, selected in the previous steps, as Jill showed you, and the population structure, which has to be externally calculated. And this is why we are going to provide very soon a workflow to calculate the population structure information. And once you have all your data here, you can select your files here from your laptop or here from the previous steps. You click Initiate Analysis Job, and you will obtain the summary statistics that you can use for, you can use it as it is, or you can use it for perform the meta-analysis.